Hello, welcome back to KIC'S tutorials. In our last class or in previous classes, we talked about monohybrid inheritance, that is inheritance that concerns one pair of contrasting characters, just a single pair. And we talked about the monohybrid ratio and the modifications to that monohybrid ratio. And I told you about the 1:2:1 phenotypic ratio and what causes it, which is either incomplete dominance or codominance. And then we talked about the 2:1 phenotypic ratio, that's the modified 3:1 ratio, which gives 2:1 phenotypic ratio, which is caused by lethal alleles, that is alleles that cause the death of the carrier of those alleles, that in the homozygous dominant condition, mostly in the homozygous condition, but in the heterozygous condition they can cause severe developmental anomalies. So in this class we'll be talking about dihybrid inheritance, but before we start, stay tuned. Welcome back. Now I told you in this class we'll be talking about dihybrid inheritance, and these are the headings I'll be discussing dihybrid inheritance on. Okay, these are the headings: Mendel's dihybrid cross, the dihybrid ratio as both the phenotypic and genotypic, but more focus will be on the phenotypic ratio. Okay, I'll also talk about the law of independent assortment that was proposed by Mendel, and we'll be talking about the mechanism of independent assortment in the pea plant, which is the plant Mendel used for his dihybrid cross. Okay, then let's start with Mendel's dihybrid cross. Please permit me, for the sake of the small board, to erase others, but we'll still take them and I'll write them as I'm talking about them. Okay, Mendel's dihybrid cross. First of all, before Mendel proposed the first law of inheritance, which is the law of segregation, he carried out monohybrid crosses, or a monohybrid cross, which involved parents with a single pair of contrasting characters. Now, the hybrids that were produced from that cross were called monohybrids. That is, they resulted from parents with a single pair of contrasting characters. Now, such a cross is called a monohybrid cross since it produces monohybrids. Now, that helped him formulate the first law of inheritance, which is the law of segregation of genes or alleles, which states that alleles segregate during gametogenesis, as the formation of gametes and later combine during fertilization. Now, Mendel was just trying to find out the behavior of a single trait, the pattern of inheritance of a single trait. Now, for the dihybrid inheritance, he carried out dihybrid crosses to find out the behavior of different characters in relation to each other when it comes to their inheritance patterns from generation to generation. So he carried out dihybrid crosses where he used parents that differed in two pairs of contrasting characters to produce dihybrids, which are hybrids with two pairs of contrasting characters. And like I said, such a cross is referred to as a dihybrid cross because it produces dihybrids. Now, let's find out more about his cross and how it went. He crossed a pea plant, one with yellow round seeds and the other pea plant with green wrinkled seeds. What he wanted to find out is if the yellow color of seeds affects the round shape of the seed, or the green color of seed affects the inheritance of the wrinkled shape of seeds. So what he discovered that it was that in his F1 hybrids, that's the hybrids from the parents that were crossed, the individuals in the parental generation. So he noticed that the F1 hybrids all had yellow round seeds. So no green or wrinkled seeds, all yellow and round seeds. So he got those pardon me please. All the F1 individuals had yellow and round seeds. So he went further to find out if he crossed these F1 individuals, if the F2 individuals will have variations, like in the monohybrid cross. So let me show you. The parents he crossed were, let's say, this is for yellow color of seeds, this is for round seeds, green color of seeds, and wrinkled seeds. So two pairs of contrasting characters in each parent. Okay, all the F1, these are the gametes. And this. So all the F1 individuals had this. Okay, so this is what he had. That means in the F1 there were 4 types of alleles. One, let me write them down up here. The allele for yellow seeds, green seeds, round seeds, and wrinkled seeds. Okay, so these were the 4 alleles in the F1 individuals. So these alleles can come in different combinations, which I will show you as follows. Okay, this can combine with this to give you YR. That is allele for yellow seeds, allele for round seeds. And then we can have this and this. So allele for yellow seeds, and the allele for wrinkled seeds. Okay, I won't be writing it all down to save our time. We can have also this and this. That's the allele for green seeds, an allele for round seeds, and then the allele for green seeds, and the allele for wrinkled seeds. So these are what will be found in the different gametes in the F1 individuals. So when these F1 individuals are crossed, these alleles will combine to give us different combinations that are up to 16 combinations. So I'm going to illustrate that. But first, let me mention first, when he crossed these individuals, the F1 individuals showed 4 major phenotypes. They showed 4 major phenotypes. They were yellow, round seeds, which were 9 in number. They were yellow, wrinkled seeds. Remember, I'm referring to the F2 now, the offspring of the F1 hybrids. Yellow wrinkled, they were 3 in number. Green and round seeds were 3. And green and wrinkled seeds, just 1. So he got them in this ratio, and he called this the dihybrid ratio, the dihybrid phenotypic ratio. That is 9 to 3 to 3 to 1, which is the dihybrid ratio. So, in order to explain the irregularity of this ratio being different from the 3:1 ratio that he saw in the monohybrids, he formulated a law to explain this, and that law is the law of independent assortment. So I've discussed Mendel's Dihybrid Cross. While talking about this, I've also discussed the dihybrid ratio. You've known how he got the dihybrid ratio. So now, on to the law of independent assortment. So, like I said, this law explains the irregularity, that 9:3 to 1 ratio that he saw in the F2 dihybrids. So, what this law states is that in the formation, or during the formation of gametes, that is the sex cells in sexually reproducing organisms, genes do not affect each other in relation to how they are sorted into alleles, and every combination of a particular gene or alleles has an equal chance of appearing in the progeny. I'm going to show you what that means. It's trying to say that, for instance, like in the pea plant he crossed to get the F1, we have this for yellow, round, green, and wrinkled seeds. Now, this gene for seed color, for seed shape, for round seed shape, this is yellow seed color, round seed shape, green seed color, wrinkled seed shape. This does not affect the inheritance or the sorting of this into a gamete. Remember, when they are forming gametes, this will end up in just one gamete. Both cannot end up in you can't have this. They mustn't always come together. You can't have this in one allele. It has to be this or this. It cannot be the both. So for this gene, you have this allele, and for this gene, you might have this. I'm talking about in the F2 now. Okay. So now this also does not affect how this is sorted. This can end up in one allele, and this can end up in another allele, and it doesn't matter. You know, remember, in the F1, they are this way. This, so to say, from the mother or from the male parent, this from the female parent, this from the male parent, this from the female parent, assuming that the male parent is the dominant parent in this cross. Okay, so this and this, yes, occur in the F1 hybrid, but it doesn't mean that they must end up in the same allele, because this usually happens during anaphase 1 of meiosis, as the cell division for sex cells. This is from the male and this is from the female. They mustn't always end up together. They are independently sorted. They can end up in different gametes. They mustn't, maybe if this moves to the, let's say there are two poles of the cell. Let me call this the North Pole and this the South Pole. It's not compulsory that the allele from the male parent must end up at the North Pole, and the allele from the female parent must end up at the South Pole, or this end up here and this will end up here. No, they can be reversed. This can end up here, even though it's from the father, it can end up at the South Pole, and this, yes, even though it's from the mother, can end up here. So there is equal likelihood for them, for any of them being sorted into a particular allele. There is no strict rule that this one must end up in this, in this gamete. There is no strict rule to that. Okay, now I know might sound like I'm speaking in tongues. You don't really get what I'm saying. So now I'm going to go to the mechanism of independent assortment according to Mendel's cross, so that you get to know what I'm talking about. So that brings us to the next heading. Okay, so now we are going to find out how it happens. Now, to understand how this works, let's assume that these are the alleles, like we've already mentioned before. The alleles for yellow seed shape, round seeds, green seeds, and wrinkled seeds. Okay, this is the first parent, probably the male, and this is the second parent, probably the female. It doesn't matter which of these parents is male or female. We'll always get the same results. So, like I mentioned before, the F1 individuals turned out this way. And I gave you the different combinations of the alleles when these F1 individuals are crossed. So this will be this, and so now I'm going to show you the 16 combinations that these alleles can give in the F2 progeny. Okay, let's draw a Punnett square here to make it easier for us. And the female. So we have those allele combinations. This is to show that this is a gamete. Okay. So for the mother also, pardon me, my lines might not be straight, but that's not our focus. Okay. So if these two gametes were to meet, remember these are from the F1 individuals. If these two gametes were to meet, this is what you would get as the genotype of that particular F2 individual. This to this, and this to this. Okay, this to this. I'm writing it this way because dominant usually shows itself over recessive, so that is why you're seeing it this way. Okay, Y, small y, big Y, big R. Big Y, big Y, big R, small r. And second, big Y, big Y. Small r, small r. Big Y, small r. Sorry, small y. Big R, small r. Big Y, small y. Small r, small r. Okay. Big Y, small y. Big R, small r. Big Y, small y. Okay, big R, small r, big R, big R. Small y, small y. Big R, big R. Small y, small y. Big R, small r. Big Y, small y. Big R, small r. Big Y, small y. Small r, small r. Small y, small y. Big R, small r. Small y, small y. Small r, small r. So if we're to count, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. You see? So now, if we are to check for the individuals with at least one big Y and at least one big R, that is that individual must have yellow and round seeds. So now we are going to check for them. This checks out. I'm going to be using this sign. This checks. Yes. One big, at least, remember I said at least one big Y and at least one big R. So, yes, yes, no, yes, no. There's a big R, there's a big Y, no big R. Yes, big Y, big R. Big Y, big R. No big Y. Same. Yes. No. 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 So let us count the ones that checked. 1, 2, 3, 4, 5, 6, 7, 8, 9. So at least have 9. Now we are going to check the one with one big Y and maybe one small R or two small R, doesn't matter. And see how many we have. Why we are checking this is for yellow color, wrinkled seeds. Okay, now we have two big Y. Yes, this, let me use this symbol. Yes, or let me erase this so it doesn't become too ambiguous. Okay, but I'll still maintain this symbol. This, nope. Yes. No. 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 Yes. No. No. So how many do we got? 1, 2, 3. All right. Now next we are going to check the one with at least one small Y and one big R. Doesn't matter, it could be one big R or two big R, but the thing is two small Y or one small Y, right? Yes. Okay, it should be 2. Yes, should be 2. Because this is recessive, it has to come in a homozygous form to show. Okay, so then let's use this symbol this time. All right, 2 small Y, 1 big R, or 2 big R. Let's see. No, 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 no. We have it here, so we'll give this. Yes, we have it here also. We'll give this. No, no, yes, we have it here. That is it. So we have 1, 2, 3. All right. Now we're going to check for the small Y, 2 small Y, 2 small R. No, 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 no. And here we have it, just 1. Okay, we can see that this gives us the ratio Mendel had, which is the 9 to 3 to 3 to 1. So now you understand how he got this ratio. All right, I think this is it. But also, remember I mentioned that we'll be talking about the phenotypic and genotypic ratio, but more emphasis will be on the phenotypic ratio. But I'm still going to teach you the genotypic ratio. But before I do that for the dihybrid individuals or dihybrids. I'll do it for the monohybrids first, so that you get to understand how this one works. Okay, I'm happy. I know you can guess that, seeing as I'm dancing. That's because I know you've learned something many people do not know, that gives a lot of people confusion. So, and I'm happy to be the one bringing it to you. Alright, the genotypic ratio. For monohybrid crosses, we have just one, let's say the yellow seeds, just one pair of contrasting characters. The F1 is like the parents, the F1 will therefore have this. Now the F2, that is if you cross this, you will get some YY, this and this, this and this, and this. Okay, you will see that for the phenotype, you have 3 yellow colored seeds. It is 3 to 1 green colored seed. This is the phenotypic ratio. But when it comes to the genotypic ratio, that's the genotype. You are concerned about the genotype, not what you are seeing on the individual. You have this 1 homozygous yellow, 2 heterozygous yellow seeds, and 1 homozygous green seed. So this is the monohybrid, the genotypic monohybrid ratio. That's the genotypic ratio. This is it for monohybrid. Okay, then we'll move on to the dihybrid genotypic ratio. With this knowledge, we know that if you were to consider the traits singly, that is those 2 pairs of contrasting characters, consider each pair. We are going to be multiplying, so to say, the genotypic ratio of the trait for color and the genotypic ratio for the trait for shape of seeds. This is what I mean. Like we had 1, we had 2, and we had 1 green seeds. So this is what I mean. This is the genotypic ratio for the color of seeds. Now we are going to cross this out. All we are trying to find out is the different genotypes. We've seen that there are basically 4 genotypes, well, 4 phenotypes, sorry, but we want to find out the genotypes. Even though we know that this individual, yes, has yellow and round seeds, does this individual have the allele for green and wrinkled seeds? That's what we want to know with this. So we bring this for shape of seed. In the same vein as this. All right, so we're just going to be multiplying the numbers basically. 1 times 1, we have 1, and then the alleles, sorry. Okay, 1 times 2, we have 2. And then 1 times 1, we have 1. 1, y, y, r, r. And 2. 2 times 1, 2. 2 times 2, we have 4, this, and this. 2 times 1, we have 2, y, y, r, r. And 1 times 1, y, y, r, r. Numbers, have 2 here. One again times 1, have 1. If you were to go back to that Punnett Square we made, you would see that these are what you will find there. So these numbers are the ratios, the genotypic ratios. So let me write them out horizontally so that you understand. Permit me to erase this, please. Yes, so we have, remember, yes, that one was 1, 2, 1, 2, 1, yes, 1, sorry, y, y, r, 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 I, y, y. Okay, this is just what I erased that I'm rewriting there, so that we don't get to be confused when I'm writing them out, the full ratio. Okay, so the genotypic ratio for dihybrids is 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. Okay, so it is 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. If you look, it's like a palindrome. Yes, palindromes are like a word that reads the same from both ends. You read it from both ends, you get the same. You have 1 here, 2, 1, 2, and the middle one. So this is what you can use to remember that the ratio, if it's too long for you to remember, remember that it is a palindromic kind of ratio. Okay, there's something I also want to give as a bonus. Sorry for the long class, but no knowledge is waste. So there's something, a little bonus I would like to give you. That is, if you want to confirm, maybe you were asked to make a cross, you want to confirm if that dihybrid ratio is actually correct. Maybe you are doubting, maybe Mendel must have been wrong. You want to be sure. This is how to be sure. Actually, if you were to consider 2 separate monohybrid crosses, the ratios of 2 separate monohybrid crosses, their product will give you the dihybrid ratio. Okay, let me show you how. For instance, we consider this separately. That's for seed color. Have this. So the F1 are this. Then the F2 are and okay, you have 3 is to 1. You also do the same for the seed shape. You have F1, and the F2. I'm sure you understand how I got to this. Okay, that's if you want to cross this, you get this same ratio. 3 is to 1. If you were to multiply these ratios, you would have the dihybrid ratio. That is to say, this 3 is yellow seeds, okay, sorry, yellow, green, yes. I have yellow, green. Minus 3 times round, wrinkled, okay. You might not be much of a mathematician, but it's just simple mathematics. Okay, so let's go. Yellow times round, we have yellow round seeds. Three times 3 give you 9. And then we have yellow and wrinkled to give us yellow wrinkled seeds. That is 3 times 1, have 3. Now let's turn to green. Green and round. We have 1 times 3. Give us 3. And then lastly we have green and wrinkled. 1 times 1 give us 1. So if you look, you see 9 is to 3 is to 3 is to 1. That's the dihybrid ratio. Gotten it back. So that is the end of today's class. I know you've learned a whole lot today in dihybrid inheritance. So please don't forget to share this video with your friends. Hit the like button, give us a thumbs up. I know we are doing well. But also, if you've not subscribed, please don't forget to do that and hit the notification icon, that's the bell on your screen, to get updates whenever we post a new video. And in the next class, sorry, before I forget, we'll be talking about modifications to this dihybrid ratio. Sorry. Just like there were modifications to the monohybrid ratio, there are modifications to the dihybrid ratio. It is not always 9 to 3 to 3 to 1. Okay, that's where I'll be stopping today. See you in the next class.